In this session, we will watch your Pecha Kucha presentation videos. Let's start with the first presentation titled Optimizing Taro Flour for Climate Smart Food Security, NaCl Soaking Innovations.
Good day, everyone. Today I'm presenting Optimizing Taro Flour for Climate Smart Food Security, Innovation and NaCl Soaking to Reduce Oxalate Content, based on research by Putu Widya Indra Astuti and colleagues from Udayana University. Climatic shift disrupt tropical agriculture via thermal escalation and precipitation variability, forecasting 2% yield reduction by 2100. Taro's adaptability is hindered by oxalate abundance, inducing nutritional and health impediments. Taro exhibit robustness to stressor, offering diversification potential. Saline processing attenuate oxalate significantly, yielding a viable staple for resource-constrained setting. This pertains to sustenance equity as environmental pressure intensifies scarcity. Mitigating Taro's constraint bolsters dietary integrity and regional autonomy. The inquiry applies saline intervention to diminish oxalate, employing parametric variation for efficacious economical adaptation. Taro was sourced, sectioned, immersed in saline gradient, desiccated, and pulverized. A factorial paradigm with replicate facilitate evaluation of oxalate via titration. Carbohydrate analytically, residue thermally, and hue instrumentally substantiated by statistic correlation. Data reveal substantial oxalate abatement over 67% optimally, attaining safe concentration. Mineral augmentation and chromatic enhancement occurred, tempered by carbohydrate diminution. Association indicate inverse relation with visual and inorganic metric direct with starch. This makes a real difference by creating safer, more appealing taro floor, reducing health risk. It's practical for farmers with low energy use, strengthening food chains in climate vulnerable areas like Bali, and minimizing nutritional trade-off through further adjustment. This augment documentation protocol shapes adaptive strategies and align with enduring developmental aims. In summary, this research transformed Taro into climate smart staple via sample innovation. Thank you for watching. For question or detail, reach out. Let's build a more secure food future together.
For the next presentation session entitled Climate Smart Adaptation Strategies of Impari Nutrizing Rice Farmers in Bantul, Indonesia. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hello everyone. Today we are going to share our research climate smart adaptation strategies of Impari nutrishing rice farmer in Bantul, Indonesia. My name is Riza and together with my colleagues Lestari, Zud and Cahyo, we are from the Department of Agribusiness, Muhammadiyah University of Yogyakarta. The main problem in this study is the impact of climate change on rice farming in Imogiri, Bantul. Irregular rainfall and long dry season constructs resulting in crop failure and reducing in nutrient rice productivity. Farmers also have difficulty determining the right planting timing due to uncertain climate. The opportunity is to increase the ability and resilience of farmers in adapting to climate change with the development of agricultural technology and efficient innovation using smart farming, strengthening farmer group as a forum for sharing information for farmers, and increasing counseling and training capacity for farmers to educate. This is important because climate change directly affects food security and farmers' welfare. Understanding how farmers adapt and what factors affect it helps keep rice production stable, especially for empowering nutrient rice, which support national efforts in improving nutrition and preventing stunting. This research addresses the problem by analyzing how empowering nutrient rice farmers adapt to climate change and by identifying the factors that influence their adaptation. The study focuses on understanding the farmers' adaptation strategies such as adjusting planting, using irrigation pumps, and applying local knowledge like Pranata Mangsa. The research using a descriptive quantitative method, data were collected through a direct interview using questionnaires supported by secondary data from agriculture institution. The data were analyzed using descriptive analysis scoring and binary logistic to determine which factors significantly influence farmers' adaptation to climate change. We found that the farmers in Imogiri are fairly adaptive to climate change. Factors that significantly influencing their adaptation include age, education level, gender, experience, land ownership status, income, land size, activeness in farmer group, and access to weather information. The finding highlight that farmers in this area are fairly adaptive, but still face challenges such as changing rainfall patterns and limit access to weather information. By understanding these adaptation behaviors, local government and agricultural institutions can design better strategy and training programs to help farmers maintain productivity despite climate change. This research contributes to the development of climate change adaptation strategy in agriculture sector, especially for rice farmers. It provides useful information for policymakers, agriculture extension service, and researchers to improve programs related to climate resilience and sustainable farming. Moreover, it serves as a reference for future study on how socio-economic factors influence farmer adaptation capacity. Thank you.
For the next presentation session entitled Optimizing Minimal Cytokinin Levels to Promote Sustainable Potato Micropropagation. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hello everyone, I'm Sadiq At Shukri, PhD student at the University of Silas Maret, Faculty of Agriculture. Our title is talking about optimizing the minimal cytokinin levels to promote sustainable potato micropropagation. The problem of this research, as we know, potatoes are playing a very important role in global food security. It's commercially propagated by vegetative part which makes them susceptible to viral and bacterial disease. And finally, it can reduce the yield and the quality of the crop. The opportunity of this study by using plant tissue culture, we can provide rapid and scalable method for producing uniform disease-free potatoes and also optimizing cytokinin levels. It can enhance plant growth while ensuring plant sustainability. The importance of this study, by optimizing cytokinin levels in potato tissue culture, we can decrease the chemical waste and improve plant resilience while supporting sustainable agriculture to meet global food demands and reducing the environmental stress. The research addressing evaluation of two potato cultivars, Granola and Bonito Agrihorti, using different concentration of cytokinins to identify the most effective combination for promoting plant growth and development. The methodology of this research, a factorial design with three replication, was conducted using MS media with different concentration of two types of cytokinins, and the growth parameter were measured after 2 and 4 weeks from the date of subculture. The result indicates that Bonito shows a better growth, while PAP at concentration 0.3 0.5 ppm promote plant growth and development. Overall, 0.5 ppm recorded the best result. The research make a big difference by optimizing cytokinin levels, promote robust adaptable potato seedlings, enhancing micropropagation efficiency. The research will contribute by supporting sustainable potato seed production system, enhancing food security and improvement potato micropropagation, and reducing environmental stress. Finally, I would like to appreciate the whole conference committee for their efforts on behalf of the authors, and thank you.
For the next presentation session entitled Ex situ conservation of wild nutmeg, Myristica spp, exhibiting superior characteristics identified through morphological markers. If you have any questions regarding the previous presentation, please kindly write them in the
Good morning, everyone. My name is Rahmat Mandir from Cairo University, Indonesia. Today, I'm honored to present our research on the conservation of wild nutmeg, which diversely focus on morphological characteristics. Now, what is the problem? Wild nutmeg population in Hanoi are genetically rich, but fully document the population face a threat from deforestation, stripping, agriculture, and mining expansion. Without proper morphological data, conservation and breeding efforts remain limited. We need to understand and protect the genetic resource before it's lost. One nutmeg offer type that are available for crop informant such as stress resistance and ecological adaptability. This research matters because while Netflix serves as a genetic reservoir for future product development, This diversity reflects biological, ecological resilience, and domestication potential. The conducted field-based morphological characterization of 64 wild nutmeg location across three ecological blocks: Wyoming, Mother Chile, and Patani. We measured 20 morphological types, including fruit, size, shape, length, dimension, and noise characteristics. We found significant morphological variation across blocks. Wyoming accession had a larger fruit and leaves, while Patani accession showed distinct shape and brilliant noise. This difference reflects both genetic diversity and environmental influences. Our findings confirm genetic diversity and ecological differentiation among not wild nut population. We identified for conservation and breeding programs. They support internal plants management and inform policies for sustainable spice development. This research provided the morphological baseline for nutmeg plants and conservation. It offers strategic input for agroforestry, environmental development, and biodiversity preservation. Thank you for your attention.
For the next presentation session entitled Assessing Genetic Diversity in Indonesian Rice Germplasm Using Start Codon Targeted SCOT Polymorphism Markers. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hi everyone, my name is Juni Widastuti. I am affiliated with the National Research and Innovation Agency and IPB University. In this presentation, I will discuss assessing genetic diversity in Indonesian rice germplasm using StatCodon targeted polymorphism markers. Indonesia has a rich collection of rice germplasm, but the genetic potential is still underexplored. Evaluation using morphology alone cannot fully reveal genetic variation. So, combining with molecular characterization provides a more complete understanding and support for breeding programs. Scott markers focus on the start codon region of gene to detect genetic differences. They are simple, low-cost, reliable, and cover a wide range of genome. Many researchers have applied Scott to identify genetically diverse accession. Why does it matter? Understanding genetic diversity is needed to broaden the genetic base of rice breeding. It helps breeders to select parental with good rates to improve genotype. Providing diverse genetic resources also support food security and strengthen adaptation to climate change. We use 15 Scott markers to assess genetic diversity and also analyze 51 genotype of rice germplasm consists of local body and varieties. This study aims to analyze the genetic diversity and relation among accession for selection of promising lines. We collected rice germplasm accession from various regions of Indonesia. Genomic DNA was extracted using the setup method, followed by PCR amplification with selected Scott primers. The polymorphic bands were scored and analyzed using Jacquard similarity and UPGMA clustering. We found that 4 Scott markers produce clear and consistent bands showing high polymorphism. A total of 112 alleles were detected, indicating strong genetic variation. The high PIC and heterozygosity confirm that the Scott markers are effective for the study. The analysis saw high kinetic variation. Most variation occur within population. UPGMA and PCA group the accession into two clusters, separating local and improved varieties. It's showing that strong genetic potential for breeding. The finding provides a molecular basis for grouping rhizoplasm and helps identify unique and diverse accession for use in breeding programs. Furthermore, it supports genetic resource management and conservation strategies at the national level. This study contributes to the molecular characterization database of Indonesia rice, thereby enhancing the utilization of genetic diversity in rice breeding. In addition, strengthens scientific collaboration in rice genomic and bioinformatics research in the future.
For the next presentation session entitled Assessing Above Ground Biomass and Carbon Stocks Among Vegetation Types in the Borneo Tropical Rainforest. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hello everyone. Today we would like to share our study about assessing above ground biomass and carbon stocks among vegetation with the authors Muhammad Hairuzaki, Umi Munawaro, and Muhammad Argahita. In this study, we start from the first question: what is the problem? Global surface temperature increased since 1850 as a function of cumulative CO2 emission. Future cumulative CO2 emission differ across scenario and determine how much warming will experience. In addition, the land use changes will exacerbate this condition. So, what are the opportunities of this study? For the first, the development sustainable land. And the second, using advanced technology such as remote sensing provide a non-destructive efficient tool to estimate above ground biomass and carbon stocks in the large area. And the last, mangrove restoration present as a natural climate solution to offset carbon deficit from land conversion. Why does it matter? Accurately assessing and managing carbon stock is critical to prevent greenhouse gas emission, climate mitigation, and contribute to Indonesia carbon neutrality targets. How is the study addressing it? The study integrates field-based carbon measurement with remote sensing data, statistical modeling to develop a predictive model of above-ground carbon stock. It also assesses potential mitigation strategies such as mangrove-based carbon sequestration to reduce carbon deficit caused. In this study, we conducted field sampling in Kabupaten Pasir and Kabupaten Penajam Pasir Utara in Borneo, collecting the data such as vegetation type, soil type, and biomass density. Also, we process satellite imagery using QGIS and make it data analysis and modeling. In this study, we found the total above ground biomass up to 352 ton per hectare and carbon stock 1,000 ton per hectare that there is to the vegetation density that we analyze using NDVI and near and R square up to 0.77. In this study, we also got the data there is a land conversion up to 46,000 hectare and making carbon loss 6.7 million tons. And in this study also, there is available mangrove area 24,000 hectare and make it offset 2.7 million tons CO2 and need more expanded area to balance the deficit. What the difference does it make from this study? This research provides a scientifically grounded quantitative method to estimate carbon stock loss, design evidence-based mitigation strategies, and inform spatial environmental policy in the development of sustainable agricultural zone, especially near IKN. The last, we hope this study contribute to climate change mitigation and adaptation framework, carbon accounting system, carbon modeling applicable to tropical landscape. Thank you.
For the next presentation session entitled The Effect of Amaranthus spinosus extract as a natural biostimulant on the growth of peanuts. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hello everyone, I'm Risa Kurnia Anggerini from Master of Agronomy Study Program Universitas 11 Maret. Today I will present our research in transforming a common agriculture problem into a potential sustainable solution with the title The Effect of Amaranthus spinosus Extract as a Natural Biostimulant on the Growth of Peanut. Peanut cultivation face significant challenge from biotic stress, particularly competition with allelopathic wheat. Conventional approach may not adequately address the complex interaction between crop and with allelochemical. Many farmers rely on synthetic input that can have environmental consequences. Allelochemical from wheat like Amaranthus spinosus can exhibit hormetic effect. This natural compound have potential as a biostimulant at specific concentration, opportunity to transform a common wheat into a valuable agricultural resource, and then a potential for developing sustainable natural growth enhanced for legume crop. Peanuts are crucial legume crop and vital source of plant-based protein. Sustainable agriculture demands eco-friendly alternative to synthetic input. Understanding variety-specific response can optimize cultivation practice. Natural biostimulant can reduce environmental impact while this research investigates the dual nature of allelochemical and then stressor versus biostimulant, and then examine a concentration depend effect across different peanut variety. Use systematic experimental design, factorial randomized complete block design to isolate variable, and then measure multiple growth parameter for comprehensive assessment. This research conducted from July to September 2025 at Universitas 11 Maret Greenhouse used two peanut variety Talam 2 and Hibama 2 with 5 extract concentration 0%, 0.5%, 1%, 1.5%, and 2%, measure 7 growth parameter and 7 week after plant. This research showed that variety had a significant effect of plant height. The Hibomatu variety reached taller than the Talam 2 variety. And then we found a significant interaction for leaf number between the variety and the extract. There is on polynomial regression. This research demonstrates that biostimulant effects are both concentration dependent and variety specific, provide scientific base for precision application of natural biostimulant, and then highlight the importance of genetic difference in response to biostimulant, and then offer practical guidance for peanut farmer in selecting variety and application rates. This research advance understanding of hormetic effect in crop with interaction and then provide evidence for variety-specific response to natural biostimulant. This research also development of target biostimulant formulation for different peanut variety and then reduction in synthetic input usage stroke natural alternative. Thank you for your attention.
For the next presentation session entitled Development of the Agricultural Sector based on an ecological footprint approach to support food security in Jombang Regency, East Java. If you have any questions regarding the previous presentation, please kindly write them in the chat.
Welcome everyone to my presentation. My name is Eko Budi Santoso. I will share insight into sustainable agriculture development in Jombang Regency using the ecological footprint approach to support food security in over the past decade, Jombang has experienced significant challenge. Approximately 6% loss of farmland annually due to land conversion for urbanization and infrastructure. Also, trend of decreasing land and management. The utilization of ecological footprint using the ecological footprint methods and data driven land management for projecting land carrying capacity and also promoting the sustainable agriculture and okay. The central question of this research is how can the ecological footprint inform sustainable agriculture development in Jombang? Uh, I set the three main objectives. First, to analyze the current land carrying capacity for agriculture. Second, to project future capacity based on current trend. And third, to formulate practical recommendation to optimize land use and enhance agriculture resilience. The core concept is the ecological footprint, comparing the land demand with biocapacity. We combine this with advanced technique, exponential smoothing for data forecasting, cellular automata, and neural net. The results show the current balance between ecological footprint and biocapacity. The graph illustrates that for the next few years, land carrying capacity remains under pressure, considering ongoing land conversion. The projection up to 2044 suggests that if current trends continue, we may face increasing overshoot, threatening food security and environmental stability. This insight highlights the urgency of implementing sustainable. From this modeling and projection, this map serves as decision-making aid, guiding where the focus conservation effort or develop new farming area and also balancing growth with preservation of land. Yeah. In summary, land overuse is a significant issue for Jombang. To sustain agriculture land management must align with ecological capacity. Thank you for your kind attention.
for the next presentation session entitled Factors that Influence the Production of Cayenne Pepper in the Highlands of Temanggung, Central Java, Indonesia. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Assalamualaikum warahmatullahi wabarakatuh. My name is Susanawati from Department of Agribusiness, Universitas Muhammadiyah Yogyakarta. Today I will present my research with title Factor that Influence the Production of Cayenne Pepper in the Highland Area of Temanggung, Indonesia. What is the problem? Chili pepper are vegetable commodity that tend to have a high level of demand. Trogomorio district is a mountain area in Temanggung, is a center of cayenne pepper production. In 2021 until 2022, the area cayenne pepper farming decreased. What are the objective? Number one, to describe the profile of cayenne pepper farmer. Number two, calculating cost, revenue, income, and profit of cayenne pepper farming. And the last, analyzing the factor that influence the production of cayenne pepper. What are the methods? Trocomulyo village was chosen as the research location. There are 147 farmers and the study used 44 farmers. The profile of cayenne pepper analyzed descriptively, the cost, revenue, income, and profit analyzed mathematically and using multiple regressing model. Profile of farmers and the study age of farmer in the range of 31 until 63 years. And the majority of farmer education is junior high school and high school graduate. You can see in this slide. And the farming experience, the experience of farmer growing cayenne pepper in the highland is mostly 5 years, but some farmer have up to 20 years. The majority of farmer in area is 1,100 until 2,000 square meters. And for a land area of 1,673 square meter, the cost incurred by farmer are 24 million, net revenue around 42 million, and profit 38 million. And then factor that significantly influence the production of cayenne pepper and the Temanggung mountain are seed and labor. But land size, organic fertilizer, chemical fertilizer, solid pesticide, liquid pesticide, and number not significantly influence. What difference does it make? The research location is in a highland area with a loss of difference in the use input and production. In highland area, market access for selling cayenne pepper may be more difficult. And the quality of the cayenne pepper is better, the taste is superior. What will it contribute to as an information material for cayenne pepper farmer? And the analysis showed that the use of solid and liquid pesticide had a negative, so not significant impact of cayenne pepper production. Farmer need to pay more attention to the standard operating procedure.
Thank you so much for your attendance and participation. We will now have a 15-minute break. The next session, session 4, will begin at 5:30 p.m. Indonesia Western Time. See you after the break.